Hello friends, and welcome to our video lecture on topic A2.2, cell structure. Our guiding questions for today, things to keep in mind while you listen to all these details. What are features common to all cells and what features are different amongst different kinds of cells? And also, how is microscopy used to investigate cell structure? Our objectives, we're going to talk about microscopy techniques, we're going to outline cell theory, we're going to state the processes of life. We're going to compare and contrast prokaryotic cells and eukaryotic cells. We're going to describe the functions of some common organelles. We're going to identify organelles in model cells, and then we're going to also talk about some atypical cell structures. Cells are a very important topic in biology because they are the basic unit of all living things. We know this through a couple different kinds of reasoning that scientists love to use. We like to use both inductive and deductive reasoning. Inductive reasoning, we're going to take some specific observations and then use those observations to make some generalizations. This guy, Robert Hooke, he looked at some cork cells under his very simple microscope a really long time ago. And he noticed that there were these cell-like chambers in the tissues. And then scientists started to look at other living samples through the microscope, and they noticed that all the stuff that they were looking at was composed of these things called cells. So they proposed that all living things were composed of cells. This is inductive reasoning, going from specific observations to make a big generalization. And then we take that generalization and we use it to write some specific hypotheses. We're pretty sure that all living things are composed of cells. Well, let's look at human cells. Humans are alive, so we should also be composed of cells. That's an example of deductive reasoning. In order to make those really good observations, to go through those processes of inductive and deductive reasoning, we need some pretty amazing tools. One of the tools that we have to study cells is microscopes. We're going to talk here about light microscopes. The next couple slides, we're going to talk about electron microscopes, light microscopes like this guy. This is what we have in our lab at school. So this one is known as a compound microscope. Compound light microscope, because there are actually two lenses. We have an objective lens here. We have an ocular lens here. We literally take some light, this light source here. The light shines up through the sample that we put on a slide on the stage. That light gets magnified here in the objective lens, and then it gets magnified here again in the ocular lens, which is why we call this a compound as opposed to a simple microscope. The total magnification of the sample is going to be the ocular lens times the objective lens. For us at school, most of our ocular lenses are 10x. Let's say that this is a 4x objective lens. We would just do 4 times 10. Would give us a total magnification of 40x power. Some of the advantages of our light microscopes, we can view our samples in color, and our samples can be living, which is pretty cool. Some of the drawbacks, the disadvantages of our light microscopes, they're not very powerful in terms of magnification, and they don't have very good resolution. We'll talk about what resolution is on the next slide. And here we're looking at electron microscopes, so using electrons instead of light. Some of the disadvantages, opposite the advantages of the light microscopes, there isn't any color, and we definitely have to kill the samples in order to get them into the electron microscopes. The advantages are opposite the disadvantages of the light microscopes. They have crazy high magnification, and their resolution is also very high. Let's talk about what that even means. What is resolution? You might notice that I've got some dots here. It's pretty easy to tell that there are 5 separate distinct dots here. As the dots get smaller, I'm reducing the font size. This is font size 28. This is 14. This is 10. This is 4. This is, I don't know if you can even see that there are dots there. This is font size 1. As the dots get smaller and smaller and smaller, it's harder to tell that they are distinct, right? Like they start to blur together. You can tell that there's 5 dots here. It's super hard to tell that there are 5 distinct dots in font size number 1. This is the idea of resolution. In our light microscopes, we need to have things be pretty big in order to tell that they are separate from each other. They don't get blurred together. Our electron microscopes, crazy high resolution. We can actually see when we zoom in that there are 5 different dots in those very small spaces. We have a couple different kinds of electron microscopes. We have scanning and we have transmission. Both of them use electrons, and those electrons bombard the sample. In a scanning electron microscope, we're going to put our sample here on the bottom. Those electrons go through lots and lots of different amazing, important physics-y kinds of tools. And then the electrons are bounced off the surface of the sample. And then we have the detector here that is going to notice how the electrons bounce off the sample, and then we get a nice contour surface look at our sample. Transmission electron microscopes, instead we're going to slice our sample super, super thin. We're going to put it into the middle of our microscope. The electrons are going to go through the sample, and then we'll have some detectors down here that will show us what different densities and tissues and pieces and parts are inside our sample. Here are some pictures from a scanning electron microscope. So these are the surfaces of pollen grains. Again, we put our sample on the bottom, and then the electrons pop off of the surface and we get this lovely contoured image of the surface of our sample. And then here is one chunk of pollen in a transmission electron microscope. So we took one of these guys and we sliced it down the middle, took a super, super thin piece, put it into the middle of our microscope. The electrons went through it. We were able to get a picture of the inside. And again, this has to be a super, super thin slice in order for the electrons to be able to go through to make our picture. In addition to having light microscopes and electron microscopes, we have some things that we can do to our samples to make them a little bit easier for us to see in our microscopes. One of the things that we can do is stain our samples. Makes it a lot easier to see things because stains will stick to different substances in our cells. Methylene blue is an amazing stain that sticks very well to DNA and RNA. So if you're looking to find the messenger RNA, if you're looking to find the nucleus of a cell, you should stain it with methylene blue. Super easy to find those pieces. We also have some amazing fluorescent stains that will absorb the light that we're shining on it and then re-emit it in special colors. This image here are endothelial cells, so cells on the outsides of organs, basically the skin of organs. And these are going to show us, these fluorescent dyes show us different pieces and parts. So the blue is going to show us the DNA, and the green are microtubules. These are proteins that help with the shape of the cell. Then we also have red actin filaments also help to push things around in the cell. We can add some immunofluorescence, and this is kind of using that same idea of fluorescent stains, except now we're going to bind to very specific molecules. So instead of all the actin filaments, we can instead pick out specific molecules that we're going to call ligands. I know it kind of got lost under my picture here, so that should say ligands. So the ligand is going to be like here. The antibody sticks to it. We have these fluorophores. These are the fluorescent pieces that will shine under the different kinds of light. We can actually do this a couple different ways. We can have the antibody that has the fluorophore, or we can have an antibody and then another antibody that sticks to the first antibody that sticks to the ligand with its own fluorophore. Regardless, lots of amazing ways for us to look at different pieces and parts, turn on different colors in our different substances in the sample so we can better view all the cool stuff that's in our cells. Another technique that we've developed to look at our cells is freeze fracturing. So we freeze our samples in liquid nitrogen, so crazy fast, crazy cold, and then we literally crack the samples open with a knife. So here we've frozen a cell, and then we're using a knife to crack open the layers of the cell membrane. So we've got outer layer, inner layer of the cell membrane, the phospholipid bilayer. We are literally cracking apart the bilayer. And then we can plate the surfaces of the membrane with some carbon and platinum, put that into a scanning electron microscope, and we can see what the inside of a phospholipid bilayer looks like. This has allowed us to figure out how our membranes are even put together. Another technique that's pretty similar is cryogenic electron microscopy. Again, we're going to freeze stuff, but instead of freezing cells or tissues, now we're going to freeze just some protein that we dropped on a slide. And then we're going to scan those protein molecules. Computers are going to take all the different orientations of all the different protein molecules that are in the sample and use those different orientations, those different perspectives, to build computer-generated models, three-dimensional models of the proteins. And this allows us to really understand how the proteins are structured, which allows us to predict their functions. This particular protein in this image is called pyosin. Pyosin's pretty cool. It's used by bacteria to kill other bacteria. So these guys we have these uncontracted pieces. The UC stands for uncontracted. So we have these uncontracted proteins that are going to stick to the cell wall of another bacteria. And they go into the contracted mode, and this little piece pokes down through the cell wall, and then the bacteria, I'm going to say bleeds to death, but of course we know the bacteria don't actually have blood. But basically all their cytoplasm oozes out, water flows in, and the whole cell explodes and it's dead. And so this is how bacteria can attack each other. Pretty amazing. We've been talking about animal cells, we've been talking about pollen grains, we've been talking about bacteria cells. So all of these living things are cells. And so scientists over time have developed and added to this idea of cell theory. A couple different tenets of cell theory. All living things are composed of cells. Cells are the basic units of life. These are kind of like same idea, just different perspectives. Cells come from pre-existing cells. We can't just like pop up cells out of nothing. Cells have to go through cell division to make more cells. Single-celled organisms carry out all the same functions of life as do multicellular organisms. Interestingly, multicellular organisms will have specialized cells, so maybe not every cell carries out all the functions of life, but the organism will, with the collection of all of its cells, carry out all of those functions. We're going to talk about what those functions are on the next slide. These are those 7 life processes that all living things carry out. Homeostasis is the maintenance of some internal condition, like our temperature or our blood sugar levels. Metabolism is the sum of all of our enzyme-catalyzed chemical reactions when we digest our food and then use little pieces of food to make new pieces of us. That's metabolism. Nutrition, gaining that food that we need to do metabolism. Excretion, sometimes metabolism leads to toxic side products we don't want to keep in our bodies, so excretion is getting rid of those waste things. Growth, our cells are getting bigger or we're making more cells that our bodies can grow. We also respond to stimuli, so when the environment changes, we also need to do some things to make sure that we can stay alive. And then reproduction, making more copies of ourselves. If it's asexual reproduction and cloning, or sexual reproduction, combining our DNA with another individual's DNA to make offspring. These are those 7 processes. You might need to, in some future quiz or test, write some of these. And so noodle on some of your favorite mnemonic devices. How can you memorize these 7 different functions of life? Can you come up with a super cool mnemonic device to memorize homeostasis, metabolism, nutrition, excretion, growth, response to stimuli, and reproduction? Noodle on it. We'll talk about it in class. One of our guiding questions for today was, what do all cells have in common? We know that all living things are made of cells, and there are some things that all living things, the cells of all living things, have in common. And these are those 4. So the first one is a plasma membrane. All cells have a plasma membrane. It is the outside membrane layer of a cell. Sometimes it'll be inside a cell wall, and sometimes it's not inside a cell wall. It's going to regulate the movement of substances in and out of the cells. Semi-permeable, which means that some things can move in and out, and some things are blocked from getting across that plasma membrane. Lysis is the rupturing of the cell membrane. So sometimes if too much stuff is getting into the cell, the whole cell can explode, and that's known as lysis. We don't want that to happen. Cytoplasm is the juicy stuff inside the cell, between the cell and the nucleus. If the cell has a nucleus, it is going to be mostly water. Water is considered the universal solvent because it dissolves almost everything, but not the whole entire universe, but it dissolves a lot of stuff. One of the things that is dissolved in the water in our cytoplasm, lots of enzymes are floating around in the cytoplasm so that we can carry out those metabolic processes. All cells have some DNA. DNA carries genes, and those genes code for proteins. We use those proteins to carry out metabolic processes, other life processes. It also helps us build the structures of our cells. And then last but not least, we have ribosomes. Ribosomes are what we use to turn the DNA. Well, we don't turn the DNA into proteins. We use the code in the DNA to produce some proteins. And these four things, memorize these, are the four things that all cells have in common. We're going to talk now about how some cells are different. So prokaryotic cells we're going to look at. Pro rhymes with no. So remember that prokaryotic cells have no nucleus. We're talking about bacteria cells here. They do have a cell wall. They do have a plasma membrane. They do have some DNA. They have some ribosomes. They have some cytoplasm. They have no nucleus. What we have instead is what we call the nucleoid. So this in this transmission electron micrograph is showing us that we've got DNA here in the bacteria cell. It is not enclosed in a membrane. There's no nuclear membrane around the DNA. Instead, it's just kind of hanging out in the cytoplasm, and we call that area where the DNA is hanging out, we call that the nucleoid. So not the nucleus, but the nucleoid region is where that DNA happens to be in a bacteria cell. Here are a couple eukaryotic cells. Eu rhymes with do because eukaryotic cells do have a nucleus. Those all rhyme. Different kinds of eukaryotic cells, animals like us, plants, fungi, and protists. Protists are single-celled organisms that mostly live in ponds. We'll talk about them quite a bit. They do have a nucleus. They also have lots and lots of membrane-bound organelles. Prokaryotic cells do not have any membrane-bound organelles. They do have some organelles, but none of them have membranes wrapped around them. So animal cell here, we've got a nucleus. There's a nuclear membrane enclosing the nucleus. DNA in the nucleus. We've got some endoplasmic reticulum. We've got a cell membrane. We've got lots of cytoplasm. There are some mitochondria here. Over here we've got a plant cell. The plant cell has a cell wall, and just inside that cell wall is its plasma membrane. It's got what we call a large central vacuole. This is where the plant cell stores lots of water and then some other minerals that help it do its job. It's got a nucleus here with that nuclear membrane. There's a membrane wrapped around its DNA. In this particular slice of the plant cell, we can also see this guy is the nucleolus. Animal cells also have a nucleolus. It's just not in this exact slice of the cell. Nucleolus is where RNA ribosomes are made in the cell. This plant cell also has these crazy cool guys. These are some chloroplasts where photosynthesis occurs, and there are also some mitochondria floating around in that plant cell. Sometimes we oversimplify and we say animal cells have mitochondria and plant cells have chloroplasts. Plant cells actually have chloroplasts and mitochondria. They're amazing. Let's pause for a moment to compare and contrast eukaryotic cells and prokaryotic cells. Eukaryotic cells and prokaryotic cells. If you want to pause the video and do this on your own and then come back and check, that would be amazing. If you want to do it with me, that works too. This, of course, is what we call a Venn diagram. Super easy way to organize comparisons and contrasts. Let's start with a comparison. What do eukaryotes and prokaryotes have in common? They all have a plasma membrane. They all have a big chunk of cytoplasm inside that plasma membrane. They've got some DNA, and they also have ribosomes. Remember that ribosomes are how we use that DNA code to make proteins. Interestingly, the sizes of ribosomes are different. Prokaryotic ribosomes are what we call 70S. They're a little bit smaller. The eukaryotic ribosomes are 80S. They're a little bit bigger. We'll talk about what that S means later. But for now, just know that eukaryotic ribosomes are a little bit bigger, prokaryotic bacterial ribosomes a little bit smaller. The DNA, same kind of A's, T's, C's, and G's, except the DNA in eukaryotes is actually what we call associated with histone proteins, whereas the DNA in prokaryotes in our bacteria, we call it naked. They don't have any histone proteins. The DNA is also circular in our bacteria, but linear in our eukaryotes. And the DNA in eukaryotes is enclosed in the nucleus. No nucleus here. Instead, we have the nucleoid region, which is floating around in the cytoplasm. Some eukaryotes have a cell wall. All bacteria have cell walls. They're composed of different things. We'll talk about those different things a little bit later. Eukaryotic cells tend to be bigger because they've got so much more stuff in them. They have those membrane bound organelles like endoplasmic reticulum, Golgi apparatus, vacuoles. Whoa, if I could spell, vacuoles. We have the mighty mitochondria, chloroplasts, all kinds of cool stuff in those eukaryotic cells, not so much in the prokaryotic cells. They tend to be smaller because they don't have all that stuff. Good? Feeling good about our compare and contrast? Fabulous. Let's move on to how eukaryotic cells especially have some specializations that allow them to carry out specific jobs. Special jobs are going to require some specialized structures, some specialized organelles. We're going to talk super briefly about organelles here. There's going to be another lecture later that goes through these in quite a bit more detail. So here we've got the nucleus of a eukaryotic cell. Here again is that nucleus. Nucleus is surrounded by a double membrane. So it's actually two phospholipid bilayers surrounding the nucleus. Nucleus is going to hold on to the DNA of the cell. And again, this is only in eukaryotic cells, not in prokaryotic cells. An organelle that all cells have, both prokaryotic and eukaryotic cells have, are ribosomes. Ribosomes in eukaryotic cells only can sometimes be bound to, this is the endoplasmic reticulum, the ER, endoplasmic reticulum. That's an R. So the rough ER is rough because it has all of these ribosomes bound to it. Our bound ribosomes make proteins. The job of all ribosomes is to synthesize proteins. The bound ribosomes are usually proteins that are secreted out of the cell. So our bound ribosomes produce proteins that are usually sent out of the cell. Whereas we have also ribosomes that just kind of float around in the cytoplasm. We call those free ribosomes, and they make proteins that usually stay in the cell. So if we need polymerase to make more copies of DNA, that's going to be produced by free proteins. Two kinds of ER. I mentioned that rough ER. The rough ER is going to take the proteins that are made by the ribosomes and then transport those proteins to what we're going to look at on the next slide, the Golgi apparatus, so that we can secrete those proteins out of the cell. We also have smooth ER, no ribosomes stuck to it. It's all just membrane, lots and lots of bubbles and folds of membrane. The job of the smooth ER is to make more phospholipids, to make more membrane so that we can grow the rough ER, we can grow the plasma membrane. It also makes other kinds of lipids. And again, we'll talk about that more in another lecture. We mentioned on the last slide that the rough ER has those ribosomes. The ribosomes are going to make the proteins. The rough ER will transport those proteins to this guy, the Golgi apparatus. Interestingly, IB gets super annoyed if we just call it the Golgi. So don't call it just Golgi. Call it the Golgi apparatus, call it Golgi bodies, but do not stop at Golgi. Good? So what we're going to do is have rough ER is here. It's going to send some vesicles of proteins and membrane to the cis. I definitely just scribbled all over that, to the cis face of the Golgi apparatus. It is going to merge into the Golgi. The Golgi is going to modify those proteins, add some carbohydrates or any other pieces that are needed. The protein will get modified as it travels through the Golgi apparatus until it gets to the trans face. The trans face is the side that faces cell membrane. Vesicles pop out. They're going to get transported. That's how I remember. Trans face is the transport out of the cell, get transported as vesicles that will eventually merge. Notice how they're merging here, merging with the plasma membrane, and all the stuff, those proteins that were made by the ribosomes on the rough ER and then modified in the Golgi, get dumped out of the cell as those vesicles merge with the plasma membrane. We have here, I've been using this word vesicle quite a bit, so here are some vesicles. So vesicles are just little bubbles of membrane that have stuff in them. This is showing what we call phagocytosis. So this is going to be an immune cell, a big giant white blood cell. It is going to use some of its cytoplasmic, cytoskeletal structures to push its membrane out. That membrane is eventually going to merge, pinch off to make this vesicle. So now we have a bacteria in the vesicle. We have this other vesicle called a lysosome. The lysosome is filled with digestive enzymes. The cell is going to have the lysosome with the enzymes merge with the vesicle that has the bacteria in it. Those enzymes are going to kill and break down the bacteria so the bacteria can no longer make us sick. So this is one way that we can use vesicles and those lysosomes. This is showing us one of those protists. Remember that protists are single-celled organisms. They usually live in ponds, but they still carry out all the functions of life. They are not bacteria because they do have nuclei, but they have to carry out all the same functions that all other living things do. These guys live in ponds. There's lots of water in ponds. Water loves to move via a process we call osmosis into cells. But if this guy has too much water moving into its body, it could go through that process that we call lysis, where it explodes. We don't want it to explode. So these guys called Paramecium, they have these super cool vesicles, vacuoles, called contractile vacuoles. And they're going to push all the water into the vacuole. The vacuole gets bigger, bigger, bigger. And then they literally just squirt all the water out. And then they put more water in, and then it squirts out. And this is how they can maintain homeostasis, that lovely water balance inside their cells. It's by using these special vacuoles. Vacuoles and vesicles, almost the same thing. We just tend to call bigger ones vacuoles and smaller ones are vesicles. Another kind of vesicle is the peroxisome. When we go through metabolism, we sometimes make lots and lots of toxic side products. And one of the jobs of the peroxisome is to take care of those things, to break down the toxins. Our livers have so many of these because one of the jobs of our liver, the organ, the liver, is to break down toxic products from all over our body. Let's talk about some mitochondria and some chloroplasts. These guys are also super amazing. We humans have mitochondria. We do not have chloroplasts. Plants have both mitochondria and chloroplasts. We're going to talk a lot about these guys in our lecture on organelles, and then also when we get to our unit on metabolism. So don't stress too much about all the words for now, because we are going to come back to the idea of these guys multiple times. But what's super cool about mitochondria and chloroplasts, they have two membranes, an outer membrane and an inner membrane. They have an outer membrane and an inner membrane. In mitochondria, the inner membrane is folded. It's folded so much that's going to increase the surface area. We call those folds cristae. The cristae have lots of proteins embedded in them. One of the proteins is called ATP synthase, and the job of ATP synthase is to synthesize ATP. So of course our mitochondria are all about making lots and lots of ATP. Chloroplasts also have that outer membrane and that inner membrane, and then inside the inner membrane they have more membranes, thylakoid membranes, and they look like these little stacks of pancakes. Embedded in these thylakoid membranes is chlorophyll. And the job of chlorophyll, of course, is to absorb energy from sunlight, and then the plant cell, the chloroplast, can use that energy to do a lot of amazing processes, which eventually lead to the formation of glucose in photosynthesis. Here are a couple looks at the cytoskeleton. So cytoskeleton has these lots and lots of super cool different kinds of proteins. And those proteins just help, especially in us animal cells, to hold up the structure of the cell. They are kind of rounded, not spherical, but certainly rounded. They have some height to them. They're not just flat pancakes. And one of the reasons for the ability of this basically just bag of juicy stuff to stay nice and fluffy and tall is these protein filaments that help to hold the structure of the cell. They also help to move things around the cell, and they also help to make those, we talked about the white blood cell and how it can push its membrane out. These guys, cytoskeleton, helps to push membrane out so that they can engulf those bacteria into those vesicles that we talked about a couple slides ago. We also have these super cool things called centrioles in the centrosome. So the centrosome is this pair of centrioles and the membrane stuff, the gooey stuff that kind of sticks to it. The job of these centrioles, the centrosome, is to help to organize all of the other pieces of the cytoskeleton. When we talk about mitosis and meiosis, so the chromosomal part of cell division, we will talk a lot about spindle fibers, and spindle fibers are special microtubules that help to push and pull around those chromosomes during cell division. The centrosome is super important for making sure that those microtubules are pushing things around in the right directions. Last but not least, we've got, I probably shouldn't call these organelles. These are really more structures, not exactly organelles, because they're outside of the cell, not inside the cell. We've got cilia and flagella, and these guys both help with movement in the eukaryotic cells, and flagella is in both eukaryotic and prokaryotic. Cilia are eukaryote only. The bacteria have some structures that are similar to cilia. They look like cilia. But they don't function like cilia. So prokaryotes, bacteria, have what we call pili. Pili help bacteria to stick to stuff. Cilia in eukaryotes help the eukaryotic cells move. So cilia are for movement, pili are for attachment. Both prokaryotes and eukaryotes can have flagella also for movement. These guys have a lot of those same microtubules that we've been looking at in the cytoskeleton and the centrioles, lots of these protein fibers that help with movement. Let's look at a couple different specialized cells now. So here's Paramecium. And here is a YouTube video that we're going to watch in class because I cannot get it to play inside these recorded slideshows. So just look at the pretty picture for now, the microscope picture. We will look at this video actually in class later. But notice that there are some cilia that helps the Paramecium move. There are also cilia here in the oral groove. These cilia actually move the food particles down this food gullet, and then the food gets trapped in food vacuoles, and then there will be some lysosomes with digestive enzymes that merge with those vacuoles so that this guy can carry out nutrition. We talked about that contractile vacuole a little bit ago. Contractile vacuole is going to collect water that gets moved into the cell because the cell lives in a very aquatic environment, and it'll just squirt out all that extra water so that the cell does not rupture. Another super cool protist, so Paramecium, and then this guy, a Chlamydomonas, single-celled organisms that carry out all of the functions of life. Chlamydomonas is super cool because it is photosynthetic, so it's got this giant chloroplast that kind of wraps around most of its body. It's got some mitochondria, it's got some Golgi, it's got chloroplast, it's got cytoplasm, all the other cool stuff that the eukaryotic cells have. This guy's pretty cool because it is photosynthetic. And another single-celled organism that can carry out all of the functions of life in its one teeny tiny little cell. Let's look at animal cells like us. So one of the things that all cells have, and our animal cells have too, is that plasma membrane, lots of ER, here's mitochondria, here we have that amazing nucleus, there's a membrane wrapped around the DNA, here lots of cytoplasm and ribosomes and all the other cool stuff that animal cells have. Remember that our transmission electron micrographs, we take a super thin slice, so we have this three-dimensional cell and we slice a tiny little slice, and we're just looking at this little slice here in this picture. There's lots of stuff in the cell above this slice and lots of stuff in the cell below the slice. So we're not going to see all the organelles here because the cells are three-dimensional and this picture is very, very much two-dimensional. Here's a plant cell. We have that large central vacuole, super easy identifying piece of plant cells. It's going to store lots of water. There's the nucleus. This guy happens to also show the nucleolus where we make our ribosomes. Here we have chloroplasts, lots of mitochondria in here as well. One of the cool things that plant cells have that animal cells don't have is the cell wall. So the cell wall here, and that plasma membrane will be just inside the cell wall. Here is a fungus cell. This is actually a yeast cell. Mitochondria, we've got a nucleus, we've got some vacuoles. There is also a cell wall. It's a different kind of substance. This cell wall is chitin. The cell wall of the plant cell was cellulose. But notice that there's no chloroplasts because fungal cells are not photosynthetic. They are heterotrophs, kind of like us. They're super cool decomposers. So no chloroplasts in these fungal cells. Cell wall, yes. Chloroplast, no. And here's our last little idea for the whole entire lecture, my friends. Good job for hanging on till the end. We're going to look at some atypical cells. So cells that are weird, cells that we look at and we're like, you don't fit what we think of cells to be. They are exceptions to cell theory. The first up are these hyphae, aseptate hyphae in fungi. So these are fungal cells. These are the cells that grow underground in fungi. They are not roots, but they kind of look like roots, but they're not roots. They're hyphae. And some fungi actually have what we call aseptate hyphae. What happens is instead of having separate cells to build these underground hyphae, not root systems, these cells actually merge. And so you end up with this one giant cell. This whole thing is one cell with one plasma membrane and one cell wall that surrounds the whole entire thing. It also is what we call multinucleate. It has many nuclei, more than one. That's kind of weird that it has so many. Most cells only have one. They're also really big. Another kind of cell that is multinucleate and really big is muscle fiber in us animals. In us humans, the sartorius is the longest of our muscle cells, and these cells start at our hips and go all the way down to our knees. They can be 60 centimeters long. They're the longest muscle cells in the whole entire human body. Also multinucleate, they have many nuclei, and of course they're very long. Opposite multinucleate are anucleate cells. So anucleate cells, that prefix a means without. And so these are some cells that don't have any nuclei. These are red blood cells. Our fancy word is erythrocyte. Erythrocytes are red blood cells. These guys, when they are first growing in our bone marrow, they do have a nuclei, but then when they reach maturation, no nucleus left. So when they are released into our bloodstream, no nucleus there, anucleate. That's weird because cells have nuclei, but these guys do not. And then one last look at weird cells are these sieve tube element cells. These make up kind of like the blood vessels of plants, but plants don't have blood, so they're not blood vessels. But they're the vessels that sap moves through. So kind of like bloodstream, but sap stream. Anyhow, so these guys also are anucleate. They have just the tiniest little bit of cytoplasm left. They could not survive on their own. Instead, they have these cells called companion cells that live next to them and feed them. They are alive. They do have that cytoplasm, but they don't have much else. They have these companion cells to keep them alive instead. And my friends, we did it. We made it to the end. We talked about features common to all cells. There were those 4 things. They have plasma membranes, they have DNA, they have cytoplasm, they have ribosomes, and then, oh my gosh, so many differences. We talked about how we have microscopy to investigate cell structure. Some of our techniques, we have stains, we have fluorescence, we have electron microscopes and light microscopes. We talked about cell theory. All living things are composed of cells. We talked about those 7 life processes, and you're going to work on a mnemonic device to memorize them. We talked about prokaryotic cells that have no nucleus and no membrane-bound organelles. Eukaryotic cells do have a nucleus and other membrane-bound organelles. We talked briefly about some of those organelles. We talked about organelles in those model cells. We looked at Paramecium. We looked at Chlamydomonas. We looked at an animal cell, a plant cell, a yeast cell. Then we talked about some of those weird cells like muscles and aseptate hyphae of fungi and red blood cells and the plant cells, the phloem cells, sieve tube element cells that are barely alive, but they are. Good work today.